And how will we treat serious diseases like cancer? We are working every bit as ambitiously as the technological change of our day is profound, full of passion and curiosity for the greatest challenges of our society. We are among the most successful universities in the world. Venerated as the best technical university in Germany, we promote bold ideas and entrepreneurial flair for the future of our next generation talents.
Recording in progress.
And the future of our employers. Exceptional in teaching and research. Interdisciplinary, beyond subject boundaries. International at the highest level. Rooted in tradition and open to the world, we take the lead in competitions as well as university rankings. We research and study life sciences as we do the art of living. We are a large family of prize winners, alumni, students, and researchers. And we are a large enterprise that thinks and acts economically. With high standards and ambitious goals, we squarely face international competition and connect with people from around the world. But changing the world on a grand scale entails understanding its finest details in all its forms and colors, elements and areas of life. A task that day to day drives us to the highest levels of performance, which we are all committed to. After all, for over 150 years, we have been fulfilling an important mission to kindle the economy with the spark of science. And we continue doing that to this day. With our curiosity, our courage, our imagination, and our passionate desire to succeed,
好的，我们的活动马上就开始了，请同学们到前排就坐，然后把自己的手机呢调到静音或者飞行的模式。感谢大家的支持和配合。
On that day, I stood there, asking myself, where will my future start? Will we start from a backward glance? A page in a book? Or a discussion with friends? Sparks of inspiration surround us, waiting to be ignited. Make every day count, testing the laws of the universe. Make every night count, breaking the boundaries between disciplines. Explore the world with passion and curiosity. Challenge difficulties with confidence and cooperation. Magic happens here because this is Tsinghua. This is Shenzhen. Brimming with youth. Brimming with vigor. Pierce through dense fog and witness the wondrous depths of science.
好，我们的活动马上就要开始了，请各位同学、老师尽快落座，然后将你的手机调成静音或者飞行模式。感谢大家支持和配合。Our event is about to start. Please set your phone to silence mode or turn them off. Thank you for your collaborations and support.
Here, everything is borderless. Sing, dance, celebrate, and announce to the world that this is our time. Lift your head to the twinkling stars, eyes clear. The sea surging, the wind blowing, the waves crashing, the rocks silent. Climb, see the light, see the pride, see the bravery, see unlimited possibilities. There is where my future starts.
Distinguished guests, ladies and gentlemen, good day. Welcome to Tsinghua-TUM Talks on Advanced Healthcare Engineering. We are truly grateful to invite all of you here today. I'm Remy Wan, an assistant professor from the Institute of Biopharmaceutical and Health Engineering, Tsinghua Shenzhen International Graduate School, SIGS for short. And today, we are honored to have the online presence of the leadership from the Tsinghua University and the Technical University of Munich, TUM for short, along with our esteemed professors in audience online. Also, we're delighted to have the professor from both universities present here on site at this venue. Under the flagship partnership between the two universities, the Tsinghua-TUM Talks is designed to engage the wider campus communities. As our collaboration continues to expand and intensify, Tsinghua Shenzhen International Graduate School is extremely pleased to host this year's event. We look forward to welcoming more TUM colleagues to Shenzhen in the near future. Shenzhen has been the forefront in adhering to the principle of exploring the frontiers of health, technology, and engineering for the well-being of humanities. So I'm confident that our speakers from both sides will share their profound insights with you today. So without any further ado, I would like to introduce the Vice President of Tsinghua University, Professor Yang Bing, to give the welcome speech. Let's welcome.
Dear Vice President Wen Kongman, colleagues and students, ladies and gentlemen, it is my great pleasure to welcome you to this special gathering. Since signing the flagship partner agreement 4 years ago, our collaboration has flourished, demonstrating a shared commitment to academic excellence and interdisciplinary innovation. We are proud to see Shenzhen International Graduate School taking the lead on Tsinghua's behalf, driving impactful projects and fostering stronger ties between our two institutions. These efforts reflect our shared vision for a partnership that delivers meaningful contributions to global challenges. This year, we focus on the theme of advanced healthcare engineering, a field that embodies the transformative potential of integrating engineering principles with medical innovation. As healthcare evolves to address increasingly complex issues, this collaboration allows us to explore cutting-edge solutions that improve outcomes for societies worldwide. This year, we are honored to have a professor and 2 staff members from the Technical University of Munich join us in Shenzhen. Their participation underscores the importance of in-person engagement, enriching the dialogues and providing new perspectives on our shared endeavors. As we move forward, I encourage everyone here to make the most of this opportunity, engage in meaningful exchanges, share your ideas, and forge connections to sustain and expand our partnership. Together, we are shaping a future defined by innovation, collaboration, and the pursuit of solutions to critical global challenges. Thank you for being a part of this journey. I look forward to the inspiring presentations ahead. Thank you.
Thank you, Professor Yang. And next, I will pass the floor to Professor Julian Winkelmann, the Senior Vice President of the Technical University of Munich, to share her remarks. So, Professor Winkelmann.
Thank you very much, dear Vice President Yang, dear colleagues and friends at Tsinghua University. It's with great excitement that we gather here today, making another significant milestone in the Tsinghua-TUM flagship partnership. As representative of the Technical University of Munich, we extend our warmest greeting and express our gratitude for the Tsinghua and Shenzhen International Graduate School's commitment to host the annual Tsinghua-TUM Talks. Over the past 4 years, our partnership has not merely flourished, it has become a testament to our shared dedication to academic excellence and interdisciplinary innovation. We commend the Shenzhen International Graduate School for its leadership and pivotal role in driving impactful initiatives that strengthen the bonds between our two universities and foster a rich academic environment. In this year, our focus on advanced healthcare engineering allows us to harness the transformative potential of combining engineering principles with medical innovation. As we confront complex global challenges in health, our collaboration will empower us to explore pioneering solutions that enhance healthcare delivery and improve outcomes for communities around the world. And I'm happy that this year I am able to contribute to the discussion with a presentation. While I'm participating in these talks online this time, I'm thrilled to see esteemed faculty members from Tsinghua and TUM involved in the Tsinghua-TUM talks. Thank you very much, Professor Singh, Professor Zhang, Professor Ma, Professor Wang, and Professor Gerhardt. Your involvement underscores the importance of direct interaction and the invaluable insights that inspire us. So together we can foster deeper discussions and exchange diverse perspectives that will enrich our partnership, particularly as this format allows for a broader audience to engage in this important dialogue. We wholeheartedly encourage all attendees to seize this moment to engage in enriching conversations, share innovative ideas, and develop our strong connections. Through our collective efforts, we can ensure the sustainability and growth of our collaboration, which is vital for addressing some of the most pressing issues facing our global society. So thank you again for your continued commitment to this partnership. We are all looking forward to the inspiring presentation and fruitful discussions that await us today. Thank you, and the warmest regard from the Technical University of Munich.
Thank you, Professor Winkelmann. I really appreciate your remarkable remarks. And next, we're going to move to our keynote speeches. I would like to introduce the first keynote speaker today, Professor Julian Winkelmann, whose research centers around the genetic architecture and underlying molecular mechanisms of complex genetic neurological disorders and rare diseases. So, Professor Winkelmann, the stage is yours. Yeah, let's welcome.
Thank you very much. It's very an honor to present myself, and I would like to share my screen.
Okay.
Can you see the presentation?
We cannot see the slides. Yeah. Now we can see it. Yeah.
Wonderful. So thank you
Go
very
ahead,
much
please.
for giving me the opportunity to talk about healthcare through precision genomic medicines. And I would like to give you two examples on how we approach and improve genomic medicine in the future and address a disease you've maybe never heard of. That's a common complex disease, restless leg syndrome. And then in terms of rare diseases, I'm delighted to introduce you to a new initiative in Germany, how we approach the improvement of diagnosing and then at the end treating rare diseases. So first of all, I would like to bring your attention to the fact that we are dealing with two ends of the spectrum, common diseases and rare diseases. So in rare diseases, usually we have a single genetic variant, a rare variant, which is causing the phenotype, and this variant classically segregates in a family in a Mendelian fashion and explaining the entire phenotype of the family. On the other end of the spectrum, we are talking about common genetic variation, where only a single variant increase a little bit the susceptibility to a disease, and many genetic variants with a higher allele frequency are leading together with environmental factors to a complex genetic phenotype. And I would like to start with a common disease. And here I would like to introduce the phenotype to you, and that's restless leg syndrome. Restless leg syndrome is a neurological disorder, and it has an age-dependent prevalence up to 10% in the elderly. Women are twice as often affected as men. It's a behavioral phenotype. So the diagnosis so far is made by only the history of the patients. It's a very, let's say, unusual history. The patients report of an urge to move the leg, very uncomfortable and unpleasant presentations in the lower limbs. And these symptoms usually begin or worsen during resting periods. So you can imagine when individuals want to relax in the evening or during the night, they usually in these resting situations, these uncomfortable symptoms in the lower limbs start. And they're only relieved by movement. And typically these movements are worse in the evening or during night. So it interferes with falling asleep or maintaining sleep. So basically, the patients suffer from severe sleep problems initiating and maintaining sleep. But overall, the phenotypic spectrum is very broad, from individuals suffering only a few episodes in their night, and on the other end of the spectrum, severely affected individuals with daily and nightly symptoms. So there's a motor phenomenon. I just would like to introduce you to these affected individuals have repetitive movements during night, limb movements during night. And this is an example of an individual where you can see a situation. Oh, it's too loud. Sorry. You see in situation in the sleep lab during non-REM sleep, and here you see repetitive movements of the ankle and then also sometimes the knee and the hip. And these repetitive periodic limb movements, which occur during non-REM sleep in these movements, are associated with arousal reactions, so leading to a significant reduction of sleep efficiency. Well, ALS is a highly familiar trait. So half of the family members report other members being affected, so which makes it approachable to genomic medicine. And as an overview, it has a different prevalence around the globe. Here you can tell that prevalence studies point to the fact that there's a higher prevalence in the US and Canadian and also South American population. It's obviously less prominent in the Asian population, which tells us so far from studies in prevalence studies in China, Singapore, Japan, and Japan. But this is of enormous importance, this observation, and this diversity has an enormous power in dissecting genomic regions later on. To approach the disease, a classic complex genetic disease, the mode of approaching this disease are genome-wide association studies. And from early on, we have conducted this type of studies and were able to increase the number of genomic loci associated with the disease. And I would like to give you more insight into a recent study we have studied this year. It's a genome-wide meta-analysis of restless leg syndrome around the globe. In so far, we have included about 1,016 cases and more than 1,416 cases and 1.4 million controls around the globe, and also additional cases in a replication stage, and conducted a genome-wide meta-analysis. So the population included the International EU ALS Gene Consortium, cases from 23andMe, Decode Genetics, the Danish blood donor studies, interval, so and Emory US donor, so number of ALS case control populations around the globe. And this just illustrates briefly the complex study design, which was followed up with a lot of functional interpretation analysis, such as gene set pathway enrichment analysis. We investigated the tissue, the cell type, and also a number of genetic correlation, Mendelian randomization, and AQTL colocalization and fine mapping. I just would like to mention the major and most important results here, and that's the genome-wide association study, which also included the X chromosome, and also at the end included sex-specific analysis. And here we have been able to identify 164 genome-wide significant loci for RLS with a SNP-based heritability of 0.2, pointing to the fact that the vast majority of these loci are developmental factors or genetic factors which are known to be involved in early embryonic development, pointing to the fact that ALS may be a neurodevelopmental disorder where genetic factors already determining at early embryonic development an alternative wiring in the central nervous system, and later on during aging, with environmental factor, it brings about the disease. But having a closer inspection on the loci identified, I would like to pinpoint two results. So first of all, a result addressing the so-called druggable genome. What's a druggable genome? it's a set of genes that encodes existing and promising drug targets and allows researchers to identify new disease relationship for drug targets. And here, in particular, we use the functional genomic annotation of our result and combine the fine mapping approach of our loci, and we constructed a score within the genetic factor of a genomic loci identified, and then we mapped the top scoring genes against the drug of the genome and identified 13 potential candidates targeted by existing compounds. So in summary, we identified several genes that are druggable. Among them, there are glutamate receptors, and these suggest that anticonvulsants are efficient in ALS, and these should be further assessed. So, for example, there are many drugs such as lamotrigine, permapanel, which are targeting the glutamate receptor, which I identified here, and which we can directly translate to the patients. And beyond that, there's also evidence for a completely new line of treatment, such as the cholecystokinin B receptor, which is a neuropeptide receptor. Just to give you some examples among others, which are directly transferred to the patients. And second example is the polygenic risk scores, which we have used. And here on the right side, you see how we improve the polygenic risk score and using genetic variants for prediction the disease and for creating a prediction score to better diagnose the disease. And here on the right side, you see an improvement of the area under the curve by, first of all, using machine learning models, and in addition, using also other variables, so other variables and other factors, phenotypic factors, which have been included in the scores. So here we see a dramatic improvement when we use machine learning models, and all models performed best when we included, had the best performance for the next 5 years. So obviously we need further validation in different data sets, and but this is a first approach for predicting the disease, and we now can have the first tool in predicting and also preventing the disease. So before I come to rare disease, I would like to mention that we have an ongoing huge study with a collaborator at Peking University here, and this time with the medical faculty, and that's about a huge genome-wide association in restless leg syndrome, which is currently conducted in a Chinese case control study. Chen Zhao and Professor Fen are currently analyzing the data, and which shows the power of diversity, because by first approach analyzing the Chinese case control study, we already see that we have different genetic factors, and we can do a fine mapping of the loci, and a combined analysis really shows the power of working together, collaborating, and approaching this, and the power and approaching these questions and improving healthcare. So the Chinese genome-wide association studies in restless leg syndrome is currently conducted in the Chinese colleagues at Peking. So, and in my last few minutes, I would really like to change the field now. So this was the first glimpse of studies we are conducting together with our Chinese collaborators. But in my last few minutes, I would like to pinpoint and change the topic now to the field of rare disease to give you an idea of an initiative we are currently doing in Germany. So that's about rare diseases. So just imagine on rare diseases, we have an example. So this is an example of a patient which presented in our university here and in prenatal MRI already pinpointed to malformations of the limb. So the woman reported that she experienced abnormal movement of the baby, and this turned out to be an epileptic seizure. So we decided on early delivery, conducted rapid genome sequencing, and directly identified a causal variant in a known gene for epilepsy. By directly approaching a startup which was working on this on this gene, we conducted an antisense nucleotide oligonucleotide and directly targeting, and we developed the ASO directly targeting the genetic variant, and by intrathecal injection of the baby in the intensive care unit, we were able to reduce the massive seizure of the baby. But the question is, so this is an example of one single individual. How do we scale this to the population level? How do we scale the ability to identify a genetic variant, to take advantage of genomic medicine and scale this for the population level? What we really need is, besides the legal framework, the IT infrastructure, a standardized and approachable data workflow, and of course, scalable algorithm. And I just would like to pinpoint to initiative here in Germany, which is going on by law since July 1st. Whole genome sequencing is provided by the health care system. So our healthcare system is providing whole genome sequencing for 2 indications, for rare diseases as well as for cancer. And the specialty here is that universities are performing the whole genome sequencing, and they are reimbursed by health insurance when they have deposited the data in a data warehouse, and then they are reimbursed. And all the data which we have deposited in the data warehouse, they can be used for research. So all the data here, they are available for secondary research, and they can be used for research. And we have an initiative in Germany, that's German Human Genome Archive, which are the backbone, and the German Human Genome Archive is the backbone of this initiative, and this is bridging healthcare. So healthcare of one side, and on the other side, we are bridging research. So we directly combine research and healthcare. What we call is knowledge generating healthcare. So we use the clinical data for research on a daily routine to foster the innovation cycle in genomic medicine. And here, when this law came into practice, we are planning the generation of about 100,000 whole genomes in the next years, and provide raw data and process all this data. So far, we have created 6 data hubs in Germany. And here at the Technical University of Munich, we are responsible to build the genome data hub for Bavaria. So at the Institute of Human Genetics, we are currently and responsible to building the the Bavarian hub of the German Human Genome Archive, so the Bavarian data center for Germany. And to illustrate you and to give you the entire picture here, so we are building the Bavarian IT infrastructure for whole genome sequencing for medical and research as a medical and research interface. And we will be connected to the German framework and to the German network of genome hubs. And all these network, which are orchestrated by the German Human Genome Archive by itself, are connected to the European health data space. So this altogether orchestrate the genomic nets and the genomic network in the future. All this, of course, illustrates and gives us the possibility that we are sharing data and we are sharing data in the future. And I think this is the most important endeavor, that we are able to share data around the globe to improve the health of our population. And with that, I would like to conclude, and I would like to thank all participants and attendees for their participation and for participating the Tsinghua-TUM talks. And thank you very much for your attendance. At the end, I have to apologize. I have to leave a little bit earlier today, as I would like to mention that today at TUM, we also have our academic annual day. So the big annual Dies Academicus, which I have to attend myself later. Thank you very much.
Thank you so much, Professor Winkelmann. And Professor shows the best example of how to bridge the research to the clinical healthcare. So let's put our hands together to thank Professor Winkelmann again. Okay.
Thank you very much. It was a pleasure. Thank you so much.
You're welcome. You can join us anytime. And our next speaker is Professor Xing Hui. His research field covers enzyme engineering, biobreeding technology and instrumentation, synthetic biotechnology, healthcare product engineering, bioactive peptide technology, and carbohydrate drug technology. So Professor Xing, the floor is yours. Yeah, let's welcome.
Okay, Remy, thank you very much for your kind introduction. Also, it's my great honor to attend the joint talk meeting between the two universities. I think today is a very good opportunity to introduce the first part about our IBHE, the research, the activity. Then I just give 2 examples for my personal research. Okay, so our Institute of Biopharmaceutical and Health Engineering, in brief, it's IBHE. So today, I just give a big title, but very small, the story. So how can we engineer the biopharmaceutical and the nutraceutical from discovery to biomanufacturing? So I think this one, the chronic disease worldwide are becoming a serious problem in the world. I will not go to detail, and also it's a very high risk of the in China, and also take the very costly. So we need to develop the strategy for not only the prevention but also for the treatment of the diseases and also the health maintenance. So that's our the IBHE strategy. So we need the globally thinking, locally acting. So I like this word. So we need focus on the human health needs chain, so-called, from the health preservation and prevention, intervention, and the treatment of the chronic disease, and also for the therapy and the recovery of the patients with the different diseases. So our aim of IBH is to explore the frontiers of the interdisciplinary engineering science and also develop and breaking technologies for the prevention and treatment of disease and health maintenance, as mentioned. So we have basically have 4 research directions. The one is health engineering, second one is pharmaceutical engineer, third one is vaccine engineer, cell engineer. So later maybe Professor Shaohua and also Chan Yang will give the presentation for their research. So currently we have 31 PI, full-time PI. By 2035, we just give our strategy plan, the plan to the dean of the SIGS. So we will, we hope we can reach to the 60 to 70 full-time PIs. So we have a very big space for recruiting the PI from all over the world. So also for our graduate students, by 2035, we hope we can reach the student number to 1,000. So it's also quite big the number. So now also for foreign overseas faculty and students, the target will be 30% by 2035. So for the from the 4 research directions, we are initiating the so-called organized research project. We want to transform discovery to products, the production and manufacturing. So we designed 3 research research projects. The first one is by collaborating of different PIs. First one is medicine and health instrumentation and also big data engineering. I will not go detail. The second one is the green biomanufacturing of the pharmaceutical products. The third one is the smart green biomanufacturing of the healthcare products. I think all these 3 research initiatives will also we are focus on the problem orientation from the national strategy and the international frontier of the discipline, and also global collaboration, including collaborating with TUM in future, and also China and the worldwide industry demands. So we for promoting our research in IBHE, so we launched the Health Engineering Journal, this is the English journal, by supporting from the Tsinghua Press, Tsinghua Publication, Tsinghua Press. I think this journal will be a very good platform for making the exchange and also the interaction between the academic communities in the world. I really hope we can also in this journal we can do a close collaboration with TUM in future. So my research area is biochemical engineer basically. So by mainly focusing on the synthetic biology and biobreeding, and biomanufacturing, health engineer, and biopharmaceutical engineer. So today I think the representative the research achievements can be divided into 3 parts. The first one is the discovery part for the next generation. We call the heparin drugs for the treating the autoimmune diseases, as mentioned before. The second one is the high throughput mining and discovery of the functional biopeptide for developing the nutraceutical and also the pharmaceutical products. The third one is the development of the high throughput and automated biobreeding instrument and the platform technology for green biomanufacturing based on the synthetic biology. So today, due to time is limited, I just focus introduce 2 typical examples in the past 20 years. The first one is I mentioned development of next generation heparin drugs for treating the autoimmune diseases in my talk, I just use the ulcerative colitis, colitis UC as a autoimmune example. So as you may know well, the heparin is highly sulfated polysaccharide with a complex structure, and also in natural material, they are wide molecular weight distribution. So the natural resource is mainly provided from their portion small intestinal mucosa, and also their very long development history. They are more than 80 years in clinical application as a anticoagulant 这个营血啊。So but beyond the anticoagulant, actually the heparin, due to their complex structure, has a lot of the activities in addition to the anticoagulant. That's because the anticoagulant activity sequence is only taking less than 30%. In another word, more than 70% of the heparin structure that contains the non-anticoagulant sequences with a potential multifunction, including anti-infection, the anti-cancer, the anti-inflammation, etc. So due to the heparin, the drug development only based on the anticoagulation, the activity. So it's very difficult to develop the non-anticoagulant the function based on the non-anticoagulant sequences due to the breakage during the production of the anticoagulant heparin. So the key point is how can we discover or rapidly discover the new function of the heparin for the next generation the heparin drugs. So one example is incurable digestive disease, that's UC. So UC, as you may not well, it's quite difficult. They are chronic inflammatory bowel disease (IBD), and also it's a modern refractory and autoimmune disease. So their pathogenesis is unknown, and also the pathology is complex. So currently there's a very limited treatment options. That's due to this disease is very difficult, including the barrier disruption, immune response, and also the blockade self-renewal. So this is the problem for the vicious cycle of UC. As you know, in past the 70 decades, the publication related to UC increased very rapidly. So especially in recent 10 years. So that means this area is still difficult area, and the drug innovation has an important social significance and also economical the value. So how can we find the non-anticoagulant function of the heparin? So in this case, we use UC as an example. Want to discover the UC treating heparin new functions. So as I mentioned, the anticoagulant function of heparin basically hence the heparin non-anticoagulant function discovery. I think this is a very difficult problem. So we changed our strategy. So if we can construct a library of non-anticoagulant heparin derivatives without the breakage of the heparin main structure, so maybe if we can remove the anticoagulant the activity, then maybe we can keep the non-anticoagulant the function of the molecules. So we did a lot of the work in the past 10 years. The first one is we built a very big library of the non-anticoagulant heparins. Then we did the drug screening and mechanism analysis by using model mice and also the patient-derived colonic organoids, so from patients. Then we after that we identified the one very useful and powerful non-anticoagulant non-molecular heparin we call the NALHP. That molecular weight will be arranged to 5 to 6 kilodalton. So then we established a very stable and the scalable production process for this NALHP. I think this is how the story. So we found that the non-anticoagulant hybrid library actually is a very, very essential platform for especially for discovery and also for exploration of the hybrid new function in terms of the UC treatment. So they makes a long story short. So we finally we found the mechanism of the NLTP is for the UC treatment is quite complex. The first one is if we use NLTP for UC treatment, it can restore the crypt structure and function, especially the stem cell redifferentiation. Then after that, it can enhance the repairing after the epithelial barrier injury during UC the occurrence. Then lastly, by the repair process, then it can strongly inhibit the immune response due to the UC the damage. I think this is very important the discovery and also new finding, especially in this case, it's restoring the crypt structure and the function, the regeneration. I think this is very important. Then it can rebalance the immune, the homeostasis, and also the accelerate the intestinal, the epithelium, the barrier regeneration. I think these two is basically the very important. So based on this one, now we are in the very soon stage to capable of elucidating the NLTP targeting receptors. So especially in the epithelial barrier, the tissue. So this is the mechanism, the discretion. So now we are identifying the ligand sequence by the synthesis of the target NLTP fragment. So then we developed the successfully bifunction of the targeted nanomedicine. So designed based on the NLTP immunity and also epithelial barrier interaction, the principle. So now this research is ready for translation. I hope next time we can introduce some of the translational research activity. So second part is the development process, the history of our research group to develop the high throughput and automated biobreeding instrument and the platform technology for green biomanufacturing. As you know, the breeding of the microbial cell or cell factories is a core for green biomanufacturing, especially in the upstream process. We need a powerful cell factory. So meeting their process viewpoint, their efficiency viewpoints, I will not go to detail. So the biggest challenge will be how can we efficiently mine the biological dark matters. At the same time, we can engineer the cell factory with a high capability or the high the power. So our group, including my Beijing laboratory, in the past 20 years, we concentrated on the development of the so-called smart high throughput, the biobreeding technology and the instrument for the biomanufacturing. So our purpose is to enhance the high throughput mine the biological dark matters by the genotype phenotype association, the genome-wide. So key is how can we connect the genotype and the phenotype? They using the high throughput the GPA, in short. So then we can reach high throughput mining of the genome-wide functional genes. At the same time, we can reach the high throughput phenotype identification. At the same time, the evaluation during the scale-up processes. So then after we get the data between the GPA, so then we can precisely we can engineer a cell factory. So so we in the past the 20 years we have succeeded in development of the instrument, including the high throughput mutagenesis, the HARTP, atmospheric and room temperature plasma. It's a plasma technology, very powerful. And also then we developed the microdroplet-based high throughput, the adaptive evolution, and also the high throughput single cell screening and the evaluation based on the microdroplet. So all this system show the high throughput automation and phenotype online detection, and also the wide application and the low consumption and low cost and high safety. So this we also co-founded with successful compared to one company called the T-Max Tree. So now industrialized all the instrument from the ARTP to the microdroplet systems, their machine systems for different purposes, including the adaptive evolution, including the high throughput, the ultra high throughput cell, single cell, the screening, and also the large cell, single cell, the screening with different, such as the mid cell, a dream cell, and the alpha cell for the adaptive evolution. So I will not go to detail. So just comparing with the benchmarking the products, we they invented the instrument. I think that has a very unique niche, and also this dream cell, mid cell is comparative with already the industrialized the machine from UK and France. I think all systems are more easy to handle, easy to operate the companies there, and also the cost is much cheaper than the competitive by the benchmarking the products. So we now we applied the enabling high throughput biobrooding platform in different sections of for the green biomanufacturing. Such as after the genome editing, we can combine with the adaptive evolution for different purpose, combining with the LRTP mutagenesis. Then after that, by aiding of the biosensor, then we can use the dream cell and the mid cell for single cell screening and evaluation. Then we can do their pilot demonstration. Then we can, by using the resequencing technology, we can get the GP big data to design the cell factory. I think this is unique. The platform for the biobreeding and the green biomanufacturing. At the same time, I think we also recently demonstrated that these microfluidic machines are very, very useful for the microbiome, the cultural mix study. So this is example for the we call the combinatorial cell factory breeding for bioproduction, combine the ARTP, and also we combine with ultra high throughput dream cell. Then by putting the biosensor, then we can do the sorting, high throughput sorting. After that, we can do the verification. Then we can repeat this cycle until we get the strong factory. This is another example of the construction of cell factory for protein and peptide the production. So after the genome editing, by mutation or the targeting mutation, then we can combine with the ARTP mutagenesis to get a good strain. In that case, we use the microfluidic sorting system by the aid of the protein, the biosensor. There are various protein biosensor, very effective. So then we can quickly get the cell factory capable of the producing the proteins or the peptides efficiently. So this is one example for the high throughput mutagenesis and screening of fungi. Maybe if you have some experience for fungi, the experiment, you can easily understand it's very difficult to do the high throughput breeding of fungi. So we combine with the mid cell. We can allow the fungi, the larger the fragment of the fungi, they are entrapped into the droplet, the stable droplet. Then we, by aid of the biosensor, so that we can easily get a larger library of the fungi. So we demonstrate that through the almost 600 monoclonal colonies, then we can get products title increased by almost 55% the increase. I think this is also say that it demonstrated that this kind of the technologies are useful. The next the example is the next generation microbial cell factory for the secretory protein and peptide production. We have demonstrated a lot of their products using the Corynebacterium and also the Picha cells. So for different protein and antibody, especially recently we have succeeded in the hyperproduction of the human serum albumin. So this also you can see here. So we in the fermenter we can reach around the 24 gram per liter. These are industry, almost they meet the industrial requirement. We identified one unique the mutant spot. So by engineering this mutant spot, we can this called M3. So we can so we can increase the productivity and also the titer at the same time. So as a summary, I think in our IBC, including myself, we are doing the AI-added disruptive innovation for the pharmaceutical and health engineering. So by from discovery to engineering. So one part that we are setting up or organizing the so-called miniaturized the high throughput biofoundry system by using our self-developed, invented the machines. And also the later Shaohua will introduce a synthetic organoid factory, so we can do the miniaturized high throughput synthetic organoid system to do the functional screening and evaluation. We hope we can combine these two parts together, then to contribute to the green biomanufacturing of the for contribution to the biopharmaceutical and the health care products development. Finally, I would like to sincerely thank to my collaborators in Tsinghua and also out of Tsinghua, and also sincere thanks for the grant support, the foundation agency to support the research. Thank you so much. Okay, thank you.
Thank you, Professor Xing, for the impressive talk. And Professor Xing, I think, showing the best research chain from the drug discovery to biobreeding and to green manufacturing. So thank you, Professor Xing. So our next speaker is Professor Markus Gerhardt, our good friend. I think Professor Gerhardt visited Shenzhen, visits SIGS in April this year. And he always showed the greatest support for the collaboration between the two universities. And Professor Gerhardt investigated the mechanism that suppressed the immune response to the host to allow the establishment of chronic infections as well as the inflammatory factors that involve in the development of cancer. So let's welcome Professor Gerhardt with warmest applause.
Do I have to switch to the slides or? Yeah, thanks. Let me start with a great thank you for the invitation and the opportunities to speak here. And I'm honored to be in your advisory board and see the progress of the Institute, and again, getting more insight today. And I hope this is going to be the basis for a lot of discussion and future interactions between our labs, departments, and universities. So I would like to take you on an excursus on our work and summing up our insights from the last, let's say, 20 years of research we have conducted in this area. My COA statement, and I'm also showing some unpublished data. And the focus of our research is a bacterial infection or a bacterium that actually didn't used to be an infection, but rather a symbiont, and nowadays pathobiont, that's called Helicobacter pylori. And it is actually one of our oldest friends and companies, having colonized humans since around 100,000 years. And having said that, nowadays, about 50% of the world population are infected with Helicobacter pylori. This graph at the bottom is actually from the Nobel laureate. So 50% of the people are infected, but only a minority of those, 10 to 20%, develop Alzheimer's disease, and only 1 to 2% really get sick and develop gastric cancer. That doesn't sound a lot, but if you extrapolate it to the number of people being infected, that makes actually gastric cancer the 4th most common cancer worldwide, and alone China has around 100 million cases, 500,000 cases every year. So a very frequent and very deadly disease. And you see the prevalence here of Helicobacter infection very closely correlates with the prevalence of gastric cancer, and especially here in Asia, are high prevalent regions. At the same time, gastric cancer is on the rise. So we see that every year we're having more gastric cancers. Just 2 years ago, we went above the 1 million mark per year. And while Helicobacter pylori infection seems to decline in some countries, also meanwhile, I think in Asian countries, gastric cancer is expected to rise for another generation. And that's because there's a latency between Helicobacter infection and cancer development. However, it would be easy to prevent. And this is a study conducted more than 20 years ago by a Japanese group, which is still very actual. And they looked at people having been treated for ulcer disease. So one of the sequelae of Helicobacter pylori infection, having been treating for ulcer disease, and the negatives never developed cancer, while the positives, this is free of gastric cancer, tended to develop gastric cancer if they were not treated. So we know we can prevent gastric cancer development by treatment. But the problem is antibiotic resistance. And we are seeing rising antibiotic resistance since years, and we can estimate that in less than a decade, it will be very difficult to treat people with Helicobacter pylori. And that's why we think that from today's perspective, we need a vaccine. And there have been many years of research on Helicobacter pylori vaccines, also some clinical trials, but eventually no vaccine was ever brought to the market. And we carefully looked at those studies and compared with our own studies to try to learn from those failures and understand what we could do better. And I would like to summarize in the next few slides our conclusions from this work. And the important thing is that also those promising candidates, oops, what happened now? Okay, now, yeah, I'm back. That also those promising candidates that were in the clinic eventually did not confer to a sustained protection. And I'm going to give you an example later on. And this failures have led to a waning interest of pharmaceutical industry. So today it's basically back to us in academia to pursue the approach of a gastric cancer preventive vaccine. And when starting to think about vaccine development, and that's also something we also discuss with our entrepreneurial students and groups in medicine, we have to start thinking from the end. So we have to start with what we call a target product profile. And this is the end of the product, but at the same time must be the starting point for a pharmaceutical development. So how would a target product profile for a Helicobacter vaccine look like? If we want to prevent the infection, we have to look at early childhood, because that's the time when Helicobacter is acquired. So we're not talking about a vaccine for adults, we're talking about a vaccine for children, which must be administered early in life. And a typical childhood vaccine has very high regulatory demands. We need to consider the route, always getting the wrong one. We need to consider the route of administration. And we know that systemic administration still is the best way to induce a very strong immunity. But we also learned during COVID disease that a purely systemic vaccination may prevent you from getting very sick, prevent bloodstream infections, viral infections, but it does not prevent people to get infected, and it does not prevent an infection to be spread. And that's why we couldn't break the pandemic during COVID. And the same would be true for Helicobacter pylori, which lives on the mucosal surface. So we require a mucosal immunity to really prevent a mucosal infection. And in very few of the approaches in the past, this has been used. And one of the reasons for this is that there is no mucosal adjuvant. So we have not a single adjuvant on the market that is approved for mucosal immunization. You have to think about dosing, how to administer this. For the mucosal route, of course, we need a mucosal route of administration, which probably for kids might best be sublingual or an oral route. We have to think about toxicity and tolerance data that need to be generated. And when we look at side effects, this is going to be one of the major criteria for judgment of such a vaccine. How well do the kids tolerate these vaccines? And when you look at clinical pharmacodynamics, then you need a strong preclinical database to define what are the correlates of immunity and what are the correlates of protection. And as I said, the target population would be young kids, at least below 10, optimally below 4 years. That's the window when Helicobacter pylori infection occurs. So a number of parameters to consider here. And if we now look at the clinical development status, and that's nearly all. So in all those 40 years we have known Helicobacter pylori, was discovered 84. So exactly 40 years, there has just been a handful clinical trials on vaccines, and I would want to point out the two ones which I find extremely informative to learn from. One from a Chinese biotech company, which made it into phase 3 and completed phase 3. And the other one from Novartis, one of the big vaccine companies. And if you look at the Tsinghua biotech vaccine, which was published in Lancet infectious disease a couple of years ago, this was an oral prophylactic vaccine. So they came very close to what's really needed for prophylaxis. And it was a fusion protein of a virulence factor, Helicobacter pylori, that's well known and characterized, and was fused to the heat labile toxin from E. coli. So a lot of things were done correctly here in this approach. And then they vaccinated young children and monitored them for about 6 years. And they saw some sort of protection in these children. So the infection rate significantly dropped. But the problem was that it dropped to 55% in the first year. But it went up already after the second and the third year. So the protection they achieved was relatively low. But still, it was the first trial in children at all, and it was the first time that an oral vaccination really could show protection. And still today, we're not really sure what happened after this phase 3 trial. It was completed in 2008, and then it never got market approval. So we tried to figure out what were the potential problems here. It's difficult for us to get this information when the study is conducted in China, but some things we could make up. What was that might have not convinced the authorities is the waning of protection. And from the data, the formulation was pretty unclear how the vaccine was formulated and administered. A big question mark from our perspective is cellular immune responses. They were not reported in the publication or in the patent application, so it's unclear if this vaccine really induced a cellular response. And as you will see later, this is really, really important. And the preclinical data were never published until 2015, although the study was completed in 2008. So something happened in this time, and we really don't understand what it was. So a number of parameters that might have contributed to the vaccine eventually not entering the market. Novartis was the other candidate that had a vaccine. This vaccine was a therapeutic vaccine and comprised 3 antigens from Helicobacter pylori that are very well known. They administered this vaccine by intramuscular injection, and that in our hands already was a red flag. Now the outcome was that they didn't see any difference between the control group, the placebo group, and the vaccinated group, and so the efficacy is here. You see here the positivity rates post-challenge. They're nearly the same. There was no protection, and the biggest problem was that they had spontaneous clearance. So, meaning that in the group that was not vaccinated, not even half of the participants were infected after challenge. So, if your positive control doesn't work, then it's very hard to see a vaccine effect. A number of other parameters made us worry here, and one is that the antigens that they used, they were not essential and they were not conserved. And what Helicobacter does, it's a genetically extremely diverse organism. It has the highest mutation rate among all bacteria we know, and Helicobacter readily adapts to the environment and turns genes on and off, and that's exactly what happened here. So if you look at these data, this is IL-8 secretion. IL-8 secretion is a direct effect of one of those virulence factors, and in the recovery strains from the patients received after vaccination and challenge, in the recovery strains, you can see that some of these strains completely turned off these antigens. And the same was true for another readout regarding the vacuolating toxin that was used. Also here, some of the strains lost this activity. So Helicobacter adapted to the vaccine, to the immunological pressure, by simply turning off the antigens we used for vaccination, which tells us that should be logic, but obviously here was not. We should use antigens that are conserved and that are essential. And so this brings us to the conclusions that we need to look carefully at the antigens. They should be conserved, and that was not always the case. And many of these virulence factors that are used for vaccination, not only in Helicobacter, but in many diseases, many of these virulence factors are dispensable. And if the bacteria can do without, then how would you effectively vaccinate? A very important point also in the Novartis trial, they used exclusively an intramuscular injection of the vaccine, so they could not, by definition, elicit a mucosal immune response. And in many of those studies, the cellular immune response was not regarded. And as I'll show you in a minute, in our hands, this seems to be the most important part of the immunity against Helicobacter pylori. And eventually, in a disease that is that is chronic and that is so well adapted to the host, we do have immune evasion mechanisms, and also those need to be addressed in vaccine design. So we need conserved antigens, we need adjuvants for mucosal routes, we need better predictive animal models. That's also important to be said, that in immunology, mice are not really predictive, and in many vaccine studies work in mice, but then they don't work in humans. And that means we need meaningful correlates of immunity and protection that can be used then to guide clinical trials. We went a completely different approach, and from the beginning, we sought to identify highly conserved and essential antigens from Helicobacter pylori. And this is a surfoam shaving approach that removes all surface exposed proteins from the surface of bacteria. And then we went to a quantitative mass spectrometry and in silico selection pipeline. And you can see that through the process from 1,500 different proteins of Helicobacter pylori, we only ended up with a handful, less than 5 candidates that really fulfilled all of those stringent criteria for immunization. And when we put those candidates into the mice, and this is a very standardized mouse model of Helicobacter pylori infection with a mouse adapted strain, then you can see that the antigens alone confer some protection, but not too much, and only the combination then really has a significant effect. And that's also typical here, that how Helicobacter evades or tries to evade the immunity. So you really need to attack it from different angles in order to achieve protection. And that's just the immunological controls with the respective antibody titers that are raised. Now, I've been talking a lot about mucosal immunity, and that's a field that is evolving more and more in the last years. And when talking about Helicobacter pylori and mucosal immunity, then what we mean is tissue resident cells. So T cells that are induced either by the infection or by the vaccination, that move into the tissue and that stay in the tissue, and then there build the first line of immunological barrier, or the first line of defense. And in Helicobacter pylori, interestingly, this is CD8. So we identified here a population of CD8 positive tissue resident memory cells. And what we did was that we infected mice with Helicobacter first, then we eradicated mice by antibiotic treatment, and then we reinfected the mice. And we were very surprised to see that this reinfection was not possible. So the mice were 100% protected from the first infection. And then when we isolated the stomach immune cells and performed FACS gating on those cells, the double positive for CD8 and CD103, we found a specific tissue memory population. And this tissue memory population was responsible for the protection. If we deplete this population, we completely lose protection. And by single cell sequencing, we could find a unique profile based on the gene expression of these cells. So these tissue resident cells are able to protect. And when we look at those cells in the immunization experiment, then you can see that these cells are induced to a different extent depending on the adjuvant we use. And when we correlate the presence of these cells with the colonization in the stomach, then we see an inverse correlation. So indeed, these tissue resident cells are responsible for the protection. Then on the antigen side, of course, we looked into conservation. I mentioned the importance of conservation for antigens, and we identified core epitopes in those antigens that are 100% conserved over all strains that over the years have been sequenced worldwide. So we really can identify core immunogenic epitopes that should confer protection to worldwide Helicobacter strains. And then put in all these informations together as a sort of puzzle, how to build the perfect vaccines. And the piece of the puzzle that is missing is adjuvant and formulation. So now knowing which antigens to use and which immunity we want to induce, the question is how. So how do we combine those antigens in a way that we can really elicit the right immune response? And that's the topic of a European Horizon-funded project, which we're currently running with 10 partners all over Europe, which is called VAX2MUC, so vaccines for mucosal surfaces. And we have built a lead candidate based on the conserved antigens we have taken. And this lead candidate is now undergoing extensive formulation development using different kinds of formulations that are known to induce mucosal immunity. So you can use chitosan-based nanoparticles, liposomes, lactic acid-based nanoparticles. And we use those in comparison with different antigens, small molecules, TLR receptors, RIG1 agonists, in order to induce mucosal immunity. And then the best of these candidates will go into preclinical studies and eventually into a phase one trial. Maybe one word about clinical trials. So also here we think that it will be very important to conduct these clinical trials in a way that they are not only valid in Germany or in one country, but that the data can really be expanded, because eventually we will need a global vaccine. In Germany, Helicobacter is not the biggest health threat. China would be a country where we really need this. So we are trying to build the phase 1, getting input from other agencies as well, including the Chinese regulatory agency, to make sure that the data we generate can really be used as a starting point for a global pivotal phase 2 trial. And we already have indications that for such an indication, preventing gastric cancer, an accelerated approval process might be possible. So we vote for a large global phase 2 trial that can be conducted transcontinental in different countries, involving all the regulatory authorities, and recruiting enough patients from each continent to make sure that this trial will be sufficient to apply for approval in different countries and in different geographic regions. And that, of course, is going to be a costly approach. We estimate that such a large trial will cost around 60 million euros or US dollar. And again, that is only going to be possible as a combined effort. We are pretty convinced that the European Commission will contribute its share. We are in discussion, for example, with the NIH and the WHO, but we will, of course, also need co-funding from Chinese partners in order to finance the part of the trial here in China. And we hope that this could be the first worldwide trial for such a prevalent and important bacterial infection, really to develop a joint vaccine to be brought to the market. I'm leaving the summary for you to read. Happy to provide the slides. And let me finish again with thanking for this opportunity. And I hope that these potential collaborations and synergisms we identify here during this meeting and our interactions will lead us to that path of eventually conducting joint clinical trials in order to tackle diseases and infections with such a global potential. Thank you very much.
Thank you so much, Professor Gerhard. Because Professor Gerhard, I think he brought up a very important issue, that is H. pylori-related gastrointestinal infections and its potential therapy by using vaccines. In the developed vaccine has a very good potency and effective in the animal models. So our next speaker is Professor Zhang Caiyang. His research is focused on the design and development of biological hybrid system with improved properties for advanced therapy based on the targeted drug discovery. So let's welcome Professor Zhang Caiyang.
Okay, firstly, warmly welcome our friends from TUM, and it's my honor to be here, and its opportunity is very generous for us to report some work for our young PI. So my topic is about the targeted delivery-based biohybrid system for advanced healthy therapy. Okay, so cause I'm a young PI, so firstly, please let me introduce myself briefly. I got my PhD degree in chemical engineering from my CUT and LMU, so I studied mutant for more than 1 year, and then I moved to Washington State University, and SMART belonged to MIT for the two postdoctoral training section. Beginning May 2021, I came back and joined IBHE. So that's my profile, and I focused on the advanced bi-hybrid system and drug delivery and control release. So as we all know that the healthy products are the fundamental secret for human life. And most of them are bioactive substance. There's substance are found in nature, and most of them are produced by chemical ways or by synthetic ways. Unfortunately, most of them cannot be administered by human directly due to lots of biological barriers in our body, like the IES and the blood vessel barrier and so on. So it's very difficult for us to use the bioactive substance directly, so including the small molecular protein and peptides, antibodies, nucleic acid, living cell, and probiotics. So it's very helpful for us, and we are lucky that we have the drug delivery. So use this way, we can construct the bi-hybrid system, which like DD or lift or U-bomb. And this car can bring the substance, the bioactive substance, to our destination. So the key for this targeted delivery BHS is the higher efficacy and the low side effect. So that's the key for our project. So in my lab, we are focused on the interaction between the artificial part, especially the micromolecules and the nanoparticle, and the bio part, especially the cells, microbial, nucleic acid, to explore the nano-micro regulation mechanism based on there's two sides. So we are focused to find the bioactive substance armor. So they can help us to do the formulation. Just Marcus mentioned that anything we need the functional formulation, even for the vaccine. So that's our work. So traditional delivery strategy is like the arrow try. So we design a different artificial part, and then synthesis, and then preparation, and then characterization. So in our strategy, we try to use the biosynthetic thought to design and screen the artificial part, including the micromolecular nanoparticles, which are easy design, multifunctional biocompatibility, and we use these artificial parts to modify the bi part, especially the polypeptide, nucleic acid, probiotic, living cell. So after that, we got the higher bi-hybrid system, which have improved features or properties, and more impressive the functions. So in our lab, in the past almost 4 years, we are focused on 4 parts: the polypeptide micromolecular hybrid bi-hybrid system, the probiotic BHS, the mRNA BHS, and the neutrophil BHS. Due to the time limitation, today I will report the probiotic E. coli Nielsen 1917 micromolecular bi-hybrid system. So considering the UC, Professor Xin just explain and introduce the UC to all of you. And so UC is a very heavy disease and worldwide. So I will save some time. And considering the currently treatment in clinical, maybe the probiotic is useful way. And until now, only one probiotic, E. coli Nielsen 1917, is the only one probiotic for the UC. So we are focused on these probiotics. Unfortunately, if we directly use the UC by oral, the function is very poor. The reason is that the GI tract, just now Marcus also mentioned that the gastric. So lots of them need overcome the barriers in stomach, in small intestine, and then they need to go to and be locked in colitis. So as we know that there's the pH in stomach is quite low, it's about 1.2, and there are also lots of enzyme over there. After that, after overcoming that, then there's barriers. Most of them should be local anti-colitis. So it's currently the oral probiotic formulation are low bioavailability and poor function and unclean mechanism. So we mentioned that maybe the viable count and colon equal triple E. It's the EP time ES and time EI. So after there's 3 barriers, the viable E. coli 1917 in anti-colitis is quite low. So we think about that, how to deliver the system or how to construct a function or advanced probiotic bihybrid system. So we think that we can do some surface engineering and to protect the E. coli well, and then help them overcome the stomach and the small intestine. And then after they arrive the colitis, they can release and release the live E. coli. So that's our idea. So firstly, we proposed the helmet and armor strategy for the E. coli. So we need to do that to help our E. coli. That's the 2 steps. Firstly, we use the calcium on the locked on the surface and find biomicromolecules which can help us to protect our E. coli and release them in anti-colitis. In this GI trick, we found that it's the bihybrid system, the BHS. They can show higher stability in stomach and in small intestine, and then in colitis they can release most of the live E. coli. So that's our strategy. So in case to find the biomicromolecules, we think about and found some collaborators. And finally, we are lucky we found the lignin, because the lignin is, as we all know that the lignin cannot be used by human. It's like the sweet moron. So it's a very helpful function for us to construct the BHS. So we tried a different modification. We use the amino acid. We use the cysteine, lysine, arginine, and so on. So we do the screening and found the satisfied modified lignin. We named it as CML. So we also evaluate the pKa, the cytotoxicity and biosafety in vitro and in vivo. Finally, we confirmed the satisfied lignin macromolecules. So, because that's not the key in this report, so after that, we got the CML. We need to do the engineering to get our desired BHS. So we use the LBL technology. Firstly, we use the curriculum on the surface and do the CML coating to get our CML modified or engineered E. coli. So that's the TEM imaging before CML coating, and this is the images after coating. We can find that there's a helmet and arm about 120 nanometers on the surface. We do a lot of work to in process investigation and optimization, and this part has been authorized by the China patent. After that, we produce this E. coli BHS. We do the characterization. We found that from the DLS, the particle size increased, the surface charge is a little bit slightly decreased. And we also use the EDS to check the different elements in our system. And we also use the SEM and the TEM to double confirm the successful construction of the E. coli BHS. And we also use the CML because we are also lucky that the CML is blue. So we needn't do any labor for check of these micromolecules. So in this imaging, we can find the red one is the E. coli, the blue one is the CML. We also use the FACS to do the confirmation, and almost 17% of the E. coli can be armored. It means that if we got 100 E. coli, at least 17 of them are protect well in our research. After that, we need to double check whether our BHS can successfully escape from our stomach. So we do the confirmation that if we don't do the coating, the free E. coli will be dead after 2 hours incubated with the SIF. But for our BSH, almost all of them are alive. That's the TEM images for 2 samples before and after CML coating. The morphology of the E. coli is integral. We also use the confocal to check the dead and live E. coli. And we also use the FACS to confirm that. We can find this. For the CML ECN, it's our BSH. Almost all of them are alive after 2 hour incubation. After that, our E. coli biohybrid system can escape the stomach. So it's like an armor. The army they overcome the first barrier. So the second barrier is the small intestine. So whether our BFH can break this barrier, we also do the animal experiment. We found that, and that's the result from the flow cytometry. Firstly, at 0 time point, almost all of the E. coli are viable here. It's meaning that most of them are still in the CML. It's integrated system. And different time points, 1 hour, 2 hour, 4 hour, 8 hours in small intestine, at least 85% of the E. coli are still protected in the CML. So the system can work to protect them. And we also take the take the samples from the small intestine and 8 hours, and we check there also some coat on the surface, so they can protect our E. coli well. In these figures, we can find that the red line is about the total amount of E. coli in small intestine. It's about, it's a little bit higher than 100%. It means that some E. coli are released and proliferation in small intestine. The blue one is the amount of the CML, ECN. It's at least 19 and 8 hours. It means that almost all of them are protected well. The most important is the profiles of this system in colon. So we also do the animal work to find that. And 8 hour time, we can find that at least 88% of the E. coli are released from the system. Only about 11% of the E. coli are still in the CML system. We can find that the solid red line is the total amount of the E. coli from the experiment. You can find that it's at 8 hours is about 114. It means that our system BHS can deliver deliver the E. coli to the colitis, and there's released E. coli can be proliferation well until the colitis. And the blue one, the solid blue one is the E. coli which are still protected by the CML. To understand this release delivery and proliferation profiles well, we also built a mathematical model based on the plug flow. So we prepared the release process model and the proliferation process model. There's two models are totally independent, dependent from the mathematical calculation. So we use these models to calculate the amount of the E. coli or the amount of the E. coli in CML system. It's the results are shown in this line, the red line. We can find that for the total amount of the E. coli from the calculation, it's very similar to the results from the experiment. So it means that our result and the mechanism exploration from the animal works and the mathematical model are very matched. And interesting that at a different time, we collect the feces and detected the CML in their feces. We found that after 3 days, almost more than 85% of the CML are in feces. It's meaning that see you tomorrow or see you 2 or 3 days later. So it means that our BHS system can successfully deliver the E. coli to the colitis and help them to do the proliferation. After that, we need to do the validation to check whether our system can treat or remit the UC effectively. So firstly, we do the biodistribution and found that under 24 hours, our system can increase the accumulation of E. coli and colitis 4.5 times compared with other control. The physical mix of the two compounds and the free E. coli. And we also checked the feces result and found that under 24 hours, our samples can help the E. coli localization and the colitis is almost increased to 10 times. And then we check whether the UC based on mouse model, whether the UC can be remitted well. We found that the in this group, our therapeutic treatment group, the length of the colon is similar to the health. And we also found the broken bearing are repaired. Professor Xing also mentioned that for the UC, there are three features. The first one, the berry disruption, and the GM disorders, and the immunohomeostasis is also broken. So firstly, it's like our kitchen. You know, the water is leaked from the tuber. So the first step for us is to repair the leakage. So first step, we use this formulation to repair the gut barriers. We also do the immunofluorescence and MPO. We found that our treatment group is similar to the health. The next, we confirm that our immunohomeostasis are well regulated by our formulation. We detailed checked the immunofactors and the inflammatory cells, and we found that our therapeutics can help us to do the immunohomeostasis. And we also checked that the GM, the third one, the GM are reshaped by our treatment. The help for bacteria like the AKK is increased. We'll also do the FIS and found that the bacteria, the GM is reshaped directly. And the last thing is about whether there's any relationship between the GM reshape and the immunohomeostasis. So we use the metabolism to find the relationship between immunohomeostasis and the GM reshape. So we found among 600 chemicals, we found 3 chemicals which are very key in this regulation between immunohomeostasis and the GM reshape. So conclusion, in this project, we designed and prepared a CML engineering probiotics. We also use this strategy to check whether it works on AKK. We also found it works. At least the efficacy is higher than 18%. It means that 100 of the AKK, at least 18% are protect well. So after oral administration, our system can deliver the E. coli probiotic to the colitis directly, and help for them to colonize in enter the colitis, and followed by receiving the GM and modulating the immunohomeostasis to significantly remit colitis. In our different projects, we still follow the same strategy to construct the functional biohybrid system to increase the therapeutic efficacy of the bioactive substance and reduce the side effect. So our work also focused on the clinical problems and the industry problems. As you mentioned, the vaccines, there we also do the microneedle, and we encapsulate the vaccine in the microneedle. It's easy for use, so we needn't do the surgery or IV injection. So maybe we can have more discussion about that. And many thanks to IBHE, to Professor Xing, and that's very excellent and amazing platform for the young PI. many thanks to our collaborators and many thanks to the members in my lab. The mission work is merely completed by Liu Junyu, and we are submitting the articles. And last word, Professor Xing just mentioned this health engineering, so it's free due to 2027. So Marcus, you are on mute. Okay, thank you very much.
Thank you so much, Professor Zhang Canyang. And I think Professor Zhang shows a very good proof of concept that the rational design of orally administered probiotics and its potential application in the treatment of colitis. So here comes the last speaker today, Professor Ma Shaohua. And Professor Ma's research includes the synthetic organs, the organoids in stem cell engineering, and organoids on the chip, 3D bioprinting. So let's welcome Professor Ma Shaohua.
Okay, thanks, Remy, for your kind introduction. So I'm going to talk about something different from the first talk. And the first one was focused on the genomic sequencing-based precision medicine and also drug development and also the probing the disease mechanism. So what I'm going to present and report to you is a synthetic organoid technology, also for the clinical usage and for the pharmaceutical development. So, okay, so we're taking cancer as a example. We know, like, say, in many cases, many developed world countries, so cancer is still remaining like the top and or the second leading cause of death. And there are over 20 million new cases every year and over 10 million deaths. So why is that? And one reason is like the cancer is always changing and it's very complicated case. So from patient to patient and from time to time, and cancer become very different. And another thing is like cancer is always evolving. It's evolving with time and it's evolving with the microenvironment. So it is not a single genome disease. Or most of the cases, it's not single genome disease. And statistics show that over the past decades, it shows like the targeted therapy on tumor, on cancer, has been very limited in the scale of eligibility and also benefit rates. So that inspires us, like, say, treating cancer cannot be just by sequencing and find the mutated genes and do the targeted sequence, target treatment. But of course, the sequencing is always fundamentally important. So we need to have the models, and the models of the black box, models of the complicated diseased organ we regard it as some kind of complicated entity. So in the past, since the 1970s, the PDX model has been merged and kept being developed for over 50 years and still now widely used in pharmaceutical companies in drug development. But it has limitations. Its cost quite high, and the genetic materials is contaminated by the host mice. And we need some new models to be more efficiently model these black box, these complicated entities, and the patient-derived organoids is such kind of this tool, our technology. So it is low cost compared with the PDX models. It takes much short time and much simpler to model. And also, like, it carries the genetic materials, purely the genetic materials from patient or from human beings. And it can also do the genetic editing on these models, and it follows precisely, can follow very well of the patient disease features and the patient development. So for this case, and we can foresee like it can be used in the drug development. It can also be used in future medical treatment, let's say precision or personalized medicine. But still, like, as a new technology, when it's merged in the biological labs, it's give you so much promises that it can do, and people can benefit from different aspects. But still, as a standard tool, it's still limited in terms like it's caused by its poor reproducibility. If you just do this technique, create the tools by using manual operations, and it consumes quite a lot of cells. That means it limits the production rate and spectrum. and growth is quite slow. So, and which culture is going to lose microenvironment and lose the features to recapitulate the patient, the disease organs. So we need, and it requires standardization as a standard too. So we need to have an organoid platform that is quite automated and increase the throughput production and increase the developmental rate and straight line modeling from the patient tumors, and also can be carried on for culture for a very long time, just like as simple as you use the cell lines, and you need to set up the standards and standards for evaluation and standards for creation of these tools. So based on this technique development, you can have the biobank of this organoids, you can have the data bank of this organoids, and you can have the instruments and the standard consumables based on all these things, and you will be able to use these tools for target findings and still in together with the sequencing technique, with the genetic and editing technique, have to validate these targets, and you can screen the drugs, and also you can probe the mechanism of functional drugs, and also treat the patient with the personalized precision levels. So then we say how to develop these tools, the platform. We have defined this as the organoid synthesis. So we know if you look at this map, and we know the organoids are assembled, and from the stem cells, and stem cells go through controlled differentiation, and also the cells go through the self-organization. It's quite like a reaction, chemical reaction in a pot, and you have the substrates, you have the enzymes, and you can have the reactants, and this reaction is occurring by different multiple substrates and in a pot, or multiple units in reaction. So it's quite analogous to that concept. So we call it the organoid synthesis. So the synthesis is guided by the chemical cues, and this chemical cues can be added to the transcriptional factor to the culture medium to get the differentiation and the maturation of the composing cells. But moreover, more than that, can also be guided by the physical cues like electric stimulation or mechanical stimulation, and also need to construct the complicated structures, so it will remind or will demand the structural definition of this organization that allows or motivates the development of precision patterning techniques of the composing units at different scales. So, and in that meaning, we say the organoid synthesis or synthetic organoid technology is by the structural, mechanical, and genetic and epigenetic regulations. So first, how to do the micro patterning in an automated and high-throughput manners, and we develop the microfluidics formulation technique in together synchronized with the bioprinting techniques. So that means you have the patient-derived cells, and you harvest cells from the patient tumor, from the tumor tissues, and using the cells create the bio-ink, suspending the cells in the matrigel in some kind of soluble fluids. So go infuse this bio-ink in the microfluidics module, so it quickly formulates this uniformly constructed microstructures, and each microstructure is supposed to develop into organoids, and each microstructure can accommodate or encapsulate over 10,000 cells, so it forms the cell colony. And then together it's synchronized with the bioprinting module, so it allows the manipulation of these organoids in automated manners. So you can see each particle or each microdroplet is only 50 microliter. And that's very small volume compared with the capacity you can do with your pipetting. And in each small volume, it can contain 10,000 cells. And you can also tailor the cell composition. So we see say for tumors, and it can have the tumor cells, and it also can have the microenvironmental cells like the fibroblasts and like the macrophage, and also the infiltrated T cells. So now people kill the cancer, always say kill the tumor cells. But now more and more studies trying to say I want to regulate the environmental cells or target on the fibroblasts, on the macrophage, to allow the tumor cells, to allow the T cells to circulating T cells to infiltrate the tumor tissues. So that gives you the opportunity to construct all these elements in the small structures. So, okay, you have the technique, but can't be done by everyone, performed equally good by everyone, then you will need an instrument. So that's the equipment we developed, and it has all the functions as been elaborated in the last figure, the microfluidics-based high throughput formulation module, and also the bioprinting module to allows you to precisely automatically pattern all these organoids into your high throughput culture container. And more than that, it also has another microfluidics module that allows the infusion of the bioink to the whole system in an automated manner. That means when you have the bioink suspended cells in the fluids and dock the tubing into the container, and you don't need to do anything. So it can all be performed by a controlled program. So and another thing is that it's very compatible with small volumes. So you have the biopsy tissue samples, and the tissue volume is very limited. And if you can only construct like the 5 microliter of the bioink. And normally by pipetting, if you waste half, and you can only produce like 1 joule or 1 drug evaluation unit or the organoid unit. But using this system, and it eliminates all that volume, nearly all that volume. So for the even 5 microliter the bioink, and you can still use up like say 40%, take advantage of 80% of the total volume, and creates 30 organoids, and 10 microliter nearly 100 organoids, and 20 microliter is several hundred organoids. So it's quite large in number compared with what it can do with other techniques. So, and if you keep increasing the bioink volume, it can even generate 1,000, 10,000, even 100,000 the screening units. So that can cover the many cases of the clinical scenarios. So how good are these organoids? And we can compare it with the primary tissues, and by using 2 different types. So take the liver tissue as an example. So using the healthy liver tissue is represented by the mouse liver tissue, and you can see like the mouse tissues are quite uniform across different individuals. But also for the synthetic microfluidics, synthetic organoids, they are also quite uniform, and they are quite alike with the mouse liver, and much better than the traditional manual operated liver organoids. And with the patient tumor, liver tumors, you can see the transcriptional analysis, the correlation or the coefficient is very high. It can reach like 0.8, that's already very high. Another case is like 0.6. So both dramatically improved than the traditional methods from the 0.3 or something. So, and of course, we say like say for precision medicine, and when you need to use patient samples to do the retrospective studies for hundreds of thousands of patient tumor and modeling, we have tried now and tested on a few hundred patient samples. And the screening of the evaluation of the 31 drugs and reaches like over 80% accuracy. So that study was performed a few years ago, and also you can do the predict the drug uses for different patients, like say pancreatic cancer patient and lung cancer patient, and it only takes you 90 days to create the models and screen the drugs. So compared with this over 3 months or 6 months for creating the animal models to predict the drug. So that was a work we mainly did a few years ago. But by continuously developing the platform, we want to use it in the new drug development, discovery, and development. So, and we welcome all different techniques, and like the data acquisition techniques by sequencing, and the AI technologies to mine to do the data mining. So like this one. And we use we using the clinical data, we pinpoint on one specific target, upon cancer target, and by using the AI algorithm, a transformer algorithm, and together we selected drugs from the candidate drugs from drug molecule pool library that contains over 10,000 drugs, and 10 over 10 drugs were selected by the choice, so like they have high binding affinity with the target protein. And these 10 drugs were further analyzed and screened and analyzed by the PDX models and by the online models, and also by the PDX models. And you can see like they have very good cytokinin effect But what's interesting here is that even two drugs of the same or similar cytokinin effect and targeting on the same target, and they were proven to have totally different cytokinin mechanism. So that study is not finished yet, but it suggests like say maybe in the future AI-based drug development should be discovery or development should be combined with the advanced wide lab screening and evaluation tools to really develop the new drugs. And for drug development and for probing the targets and for probing the drug functional mechanism, you also need to have the in-depth data acquisition and analysis. So for the transcriptional data acquisition, and we developed the on-noise spatial transcriptomics technology. And this one is the only technique ever reported to for the spatial transcriptomics to screen the on-noise. Is because compared with the traditional operations, you have to slice the on-noise. It's quite small, it's difficult to operate, and in each slide, it contains only a few number of cells. It doesn't give you special information doesn't give you special correlation of the composing cells, but and also it's quite costly. But this technique, we just use one-step lamination method to preserve all cells and also relatively preserve the cell-cell interaction in space. And it's very simple, very easy to operate, and also allows you to operate to process more than one on at a time to reduce the cost. So when you develop the drug, not only you need to know like say can kill the cell, but also you need to know like say this drug molecules is not toxic to human liver or like the heart. So one example is like here we use this synthetic on-noise technology can also purely in vitro to create the circadian rhythmic features in the liver and evaluate the drug toxicity that has different performance at different time points of a day. So if a drug been first and preliminarily evaluated as very toxic, and you may have the chance to choose the use like different time points of the day to use the drug to be less toxic to pass through the clinical screenings, and also they can be used as the say metabolic studies, like say whether the liver is responsible to the a drug under the alcohol treatment or something. So that's the simple models and on liver and on toxicity. But how can they be like used in the healthy organs and to study in other cases, like for new target findings? So this one is a airway on-noise healthy patients, actually from the healthy tissues of diseased patients. So we study the nicotine exposure to the airway airway cells, and they were proven to be harmful to the airway cells. And what here we use a known toxic molecules and known drugs, and the drugs being used in clinics, but trying to say prove like they can be used to find new targets, validate new targets. And we can see like say this on-noise containing the beating CD. The CD has the tubulin to support the beating, and in the CD is distributed with the nicotinic acetylcholine receptor. So this is not new, but what's new is that actually we found a new receptor subtype which was not reported before. People always regard like the subtype 7 is the most dominant receptor here, but actually it's subtype 5 more prevalent here. And this drug, people say like say it is antioxidant molecule to protect the cells, but actually here it's not. It is proven to be the competitive binding, so new mechanism and new target validation. So that's a simple on-noise and can be we further improve the complexity by say infiltrating the new elements to the system, simplified system. So this one is in combination with the Edwin's lab. We study the gut on-noise and inflammation appendices and creates the on-noise, and this on-noise was infected with the E. coli to mimic the inflammation. And without inflammation, and these T cells can coexist with the on-noise, but with the inflammation, T cells are going to attack the on-noise and break down the structures. Something interesting here is that is the functional mechanism screened by the molecular studies based on this activities, and it matches very well with the clinical sample findings here. So there are some mismatch, but in general, it matches very well. So it gives an understanding, like say, you can use a simplified model to pinpoint the pathways. And based on that, we even studied the more complicated models, like constructing the brain tumors and constructing the Alzheimer's disease models in the brain tissues. And first we use the animal models to using the animal models to construct, to first explore the triad interactions in the neuron tissues with the adaptive immune involvement, and regulate the neural activities by putting the mice, transgenic mouse, under different cultural nurture conditions, breeding conditions, enriched environment or depressed conditions, and they have different neural activities. And we found like the AD progress is largely determined on the breeding conditions. And in this process, the CD8 T cells, they play essential roles here. Under enriched environments or depression, the CD8 T cells, they change a lot in number in both the blood and the tissue lesions. And so when we manipulate the CD8 T cells by completely removing the CD8 T cells and or infiltrating, transplanting more CD8 T cells, it's very interesting to find like they both accelerate the AD progress, but we don't know the reason why. But for that, we still want to continue study this and what mechanism, what's the axis that's playing essential roles in this way. So we construct the simplified model of the human brain and of the innate immune system by infiltrating the organoids with more microglia, and the adaptive immune system by adding the CD8 T cells to the system. And so it has the three elements in the Alzheimer model. Something very interesting is like when you add this, create the Alzheimer models, you can see like the electrophysiology activity is quite like the human AD cases. And then add the microglia here and add the CD8 T cells, then we have all the three elements here. So something very interesting here is like without the microglial cells, the CD8 T cells doesn't infiltrate and doesn't respond, doesn't cause the physiological signal changes. And with the existence of the microglial cells, and CD8 T cells become very effective and causing the changes in electrophysiology. Now something here is like after all these studies, we conclude this scheme. It's like the astrocytes, they activate the microglia, and the microglia is going to be to respond to the engulfed or bat neurons and destroy the neurons. And another way is like this pathway is going to microglia going to recruit the CD8 T cells from the blood, and once the CD8 T cells reach the AD tissue lesion, and it's going to forcefully destroy the neurons and destroys the neurons, even the neurons still being allowed by study to engulf the Aβ42, the peptides, and it's going to accelerate the destroy or damage to the neurons and accelerates overall the AD progress. So that's the two pathways, and particularly like this pathway is a very good target for us to say stop the recruitment of the adaptive immune cells to the lesion. So in summary, like we developed the synthetic organoid technology, and which is given here, briefly described here, and we developed instruments, the machine, and to allow us to expand this technique, automatically treat that. And based on that, we have both the brain organoids and also brain models and the peripheral organ models, and we want to study particularly interest in the systemic modeling and manipulation intervention of on the neurons to manipulate or to manage the disease. Okay, so thanks all for that's my talk, and thank you for the opportunity.
Yeah, thank you so much, Professor Ma. Yeah, thank you for your impressive talk on the organoid synthesis and its potential clinical application. And next is about the Q&A section. So it's open to the audience, but since we're far behind the schedule, so probably 1 or 2 short questions.
Thank you. Hello, wait. Okay. Yeah. So I can have questions for any of the presenters, right?
Yes, for sure.
Okay. So I have a question for Marcus. Yeah. So yeah, I get a lot from the your talk about the brief history and the obstacles for the Filari and vaccines. So one thing I think that I'm thinking about, because we already identified that the gastric, there's a lot of the commensal microbiomes there. So I think there's maybe interactions between those microbiomes with the infection. So is there any studies talking about the differences, the dynamics following this infection, and about the interaction between the commensal bacteria, and also regarding to the immune invasion, about the immune responses with that? So yeah, could you answer that?
Yeah, but very good question. And I think it's important to point out that we've learned now with the new technologies that the stomach is not a sterile organ, and there's multiple bacteria, and of course there's interactions. So people have sequenced the microbiome from the stomach with and without Helicobacter infection, and you see a drastic change in the ecology in the stomach. But the same is even true for changes in the intestine and in the colon. So you can also find changes in the colonic microbiome composition in Helicobacter positives. And we think that, and we've also shown that there's two components. One is the immune system. So the infection raises a very strong immune response, and this immune response then already leads to changes in the microbiome. And at the same time, there is metabolic activities that also change and seem also to influence the microbiome in the stomach, but also in the lower intestine and colon. And if you eradicate Helicobacter pylori, then the flora normalizes. Interestingly, after eradication, the flora moves towards what you find in people who have never been infected with Helicobacter pylori. Yeah, so it's pretty clear that these changes depend on Helicobacter pylori infection directly. How that translates into vaccinology, I think that's a very big topic, and we are only starting to understand how microbiome may influence responsiveness to vaccines. There is very little data out there. The best data we can probably use to extrapolate is the relationship between microbiome and transplantation, and the relationship between microbiome and immunotherapy in cancer, because in both we know that you need a healthy microbiome to have a better outcome.
Yeah, thank you so much. So any other comments or questions from the audience? Okay.
I also have a question for Professor Marcus. So I remember that you mentioned that there is no advanced candidate for this vaccine, right? So since there are many emerging AI tools for protein design, I would like to hear your opinion about how can that be applied to the vaccine development for the H. pylori?
Yeah, I think that's a very good question, and indeed a field we are now starting to explore. As I mentioned, there is a very limited number of candidates, and we think it would be an interesting approach to use those candidates and combine them in a single protein approach, which comprises the most important parts of all those candidates, building a synthetic protein that can be used for vaccination. And certainly AI prediction of the immunogenicity, the folding, and the 3D structures would be very helpful here.
Okay, thank you.
Okay, thank you so much. So owing to the limited time, I'm afraid you have to close this section. And I think all the audience will have enjoyable and rewarding experience from the conversation between the professors and the audience. And next with this, I would like to bring the 5th Tsinghua-TUM talks to a close. And I truly relish the profound insight engaging talks as well as the stimulating answers and discussions presented by our speakers. So it is our hope that this discussion will contribute to the further deepening of the collaboration between the two universities in the future, particularly in the area of advanced healthcare engineering, which is a highly prominent and relevant topic on the global level. So once again, I would like to express my sincere gratitude to all the speakers today and the attentive audience. So we look forward to see you again next year. Goodbye.